Hi, this is Alex from devstories.eu. Biopython Fundamentals, Episode 12 of 18. If you have ever looked at a raw Newick file, you know it is just a dense, nested nightmare of parentheses and commas. Writing your own string parser to figure out which species is related to which is a miserable exercise. What you need is a way to load that text directly into a traversable data structure. That is exactly what Bio.Phylo provides for phylogenetic trees in Python. Bio.Phylo handles parsing, manipulating, and drawing evolutionary trees. The primary entry point is the read function. You pass it a file path and a format string, like Newick or Philo.xml. The read function consumes that file and returns a single tree object. If your file contains multiple trees, you use the parse function instead to get an iterable sequence of trees. Once you load the file, you work with two distinct components, the tree object and the clade object. The tree object represents the entire structure as a whole. It holds global metadata, such as the name of the tree and whether it is rooted or unrooted. But the actual branching data does not live directly in the tree object. It lives in clade objects. A clade represents a specific node and every descendant that branches from it. The tree object has a root attribute, which is simply the starting clade for the entire hierarchy. Every branch off that root is another clade, which contains its own subclades, cascading all the way down to the tips. Navigating this hierarchy is where the module shines. You do not write recursive loops manually. To search the structure, you call the findClades method on your tree. This method searches through all the nested branches and returns an iterable of clades matching the properties you specify. You can search by name, by branch length, or even by a custom evaluation function. If you only care about the absolute ends of the branches representing the current living species, you use the getTerminals method. This immediately returns a flat list of the leaf clades. You can also calculate structural metrics instantly. Calling the total branch length method on the tree sums up the lengths of every single branch in the entire structure, giving you the total evolutionary distance. Consider a scenario where you load a massive mammalian evolutionary tree. You want to isolate just the branch containing primates to run a localized analysis. You call findClades and search for the target name primates. This returns the specific clade object representing the common ancestor of all primates. Because a clade naturally contains all its descendants, you now have a complete isolated subtree. You can pass that primate clade bing to other analysis functions exactly as you would a full tree. Sometimes you just need to look at the structure to verify your extraction worked. Bio.Phylo includes a draw ASCII function. You pass your tree or your isolated clade to this function, and it prints a plain text representation directly to your terminal. It's not meant for publication, but it gives you immediate visual feedback on the topology without requiring you to configure external plotting libraries. Here is the key insight. Translating nested brackets into native objects means topology becomes an API, allowing you to slice evolutionary histories with standard method calls. If you found this episode helpful and want to support the show, you can search for DevStoriesEU on Patreon. That's all for this one. Thanks for listening, and keep building.